about protein structure. So I already showed you what an amino acid looks like, okay? And how do we make a polymer, a polypeptide? We put a bunch of amino acids together, right? And this is kind of the example of we have amino acids, all of them together, okay? We join them via dehydration reactions. You make a chain, right? An amino acid chain. So this is going to be your chain, a bunch of amino acids together, okay? So protein, now we'll talk about what happens then. And think about it like this. We're talking about that. Remember, we're talking about that necklace you're buying for your girlfriend or your mom or your grandma, right? Made of pearls. Now, if you're a student, probably you have to go to Walmart and get something, not not pearl-like, something that looks like a pearl, but it's not a pearl. Okay, so the point of the fact is you have a necklace, right? And that is like if you can expand it, okay, it's like a chain, right? But then what happens? Then you put it away, right? You put it away, and it's not going to be like a line, right? It's going to be like all intertwined. It's going to intertwine all by itself, right? Okay, so think of it like that um, as we're doing folding of this protein, because eventually it's all gonna come together. The all amino acids are going to be interacting with each other. Okay, so protein structure. So the shape of a protein, very important, is very the shape of a protein is very, 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 very important. Okay, why? And this is kind of a three-star concept here, because shape determines function. Depending on its shape, the protein can be extremely long, strong, okay, can make something like keratin, for example, could be a structural protein. Depending on the shape, a little more globular, okay, a little more like bigger and circular, okay, is probably going to be an enzyme, okay. So that is it's very, very important, because depending on how this polypeptide falls on itself is going to have a specific function. So remember that this is a very, very important concept. Okay. All right. So the order of amino acids is going to affect how the protein folds, right? Protein folding is going to determine the function of this protein. Okay. And the protein, and this is another three-star concept, okay? The function, proteins can only function if in the right conformation. It has to have the right conformation to have the specific function. And again, the reason how or why the order of amino acids is going to affect the way the protein folds, it has to do with the properties of the amino acids. Some of them are going to be hydrophobic, some of them hydrophilic, some of them are going to be polarized, either positive, negative charge, and that's how the whole protein is going to have to be able to fold, and then be able to fold the right way to be to work, to meet its function. Okay. Now there's four levels of protein structure. We have primary structure, secondary structure, tertiary structure, and in many times and sometimes, not always, you have something called a quaternary structure. Okay, so primary structure. The primary structure of a protein, of a peptide chain, is nothing but the sequence of amino acids. That's it. Okay, and this primary structure is important because it's going to determine all other levels of protein structure. So if I give you a couple of monomers here, a couple of amino acids, I'm going to draw you several of them just so you get an idea. Okay, these are a bunch of amino acids, right? And these amino acids are going to go through what? Dehydration reactions, right? So here we come, it's a monomer, an amino acid, amino acid, amino acid. You have 1, 2, 3, 4, 5, 6, 7. What is its primary structure? The primary structure of these amino acids, or the way they attach to each other, is simply this. It's just a necklace. 1, 2, 3, 4, 5, 6, 7, right? Because we went through dehydration reactions, right? we get this. This is a polymer. This is a polypeptide chain. Okay, but this is the primary structure as well. Primary. Sorry, that was a little messy. The primary structure. It's just a chain of amino acids. So far, so good. Now, next, we're going to go to the secondary structure, which is going to be based on the shape of this polypeptide. And here we go. Secondary structure is going to be the folding of the amino acid chains. Okay, the folding of the polypeptide chain. But the secondary structure, what leads to this secondary structure is a bunch of hydrogen bonds. It's the hydrogen bonding, okay, between the backbone of the polypeptide chain. Okay, when we talk about the backbone, let me go back a couple of slides so I can show you the backbone. Okay, I'm going to show you really quick just the backbone. What is the backbone of this very small polypeptide chain over here? What is the backbone? Let's do a fancy color of red. This is the backbone. Okay, that is going to be the backbone. Okay, what are we missing from here with amino acids? The R groups, right? But this is not part of the backbone. Okay, the backbone is that carbon and the amino group has amino end, the carboxyl end has the carbon in the middle. That's going to be the backbone of your of your polypeptide chain. So now we go over here. Okay, we're going to see that secondary structure is based on the polypeptide backbone. Okay, a bunch of hydrogen bonds. And what they're gonna do is they're gonna stabilize and they're gonna make this structure very stable. Okay, they're gonna stabilize the folding of the polypeptide. Now there's two main shapes. Okay, when you hear secondary structure for proteins, you're gonna be thinking there's gonna be two main shapes. Okay, the alpha helix and the beta pleated sheet. Okay, if you look here, the alpha helix here, and this is for proteins. This is a secondary structure. Okay, secondary structure. If you see how the backbone, all of these things here, these are going to be the hydrogen bonds. See, hydrogen bonds, hydrogen bonds. All of these are going to be the hydrogen bonds. All these dotted lines are the hydrogen bonds. Okay. Okay, I'm going to do this here, hydrogen bonds. And if you notice, they're going to be due to the interactions of the backbone. Okay, in order for there to be hydrogen bonds, it has to be within those hydrogens, for example, that are hanging out with other oxygen molecules, for example, in the other backbone. Okay, but the shapes itself, they're going to be making two shapes: the alpha helix, okay, and the beta pleated sheet. Okay, this is another secondary structure. Okay, I'm not gonna erase this, but I'm just gonna rewrite it. Secondary structure, okay? It could be either alpha helix or it could be a beta pleated sheet. And if you're gonna look and just take this picture, it's not very accurate, it's not very clean, but I'm gonna draw a polypeptide. This time, I'm going to draw it like a line, okay? Just to make it easier. Okay, that's a primary structure. Now let's go to a secondary structure. It's gonna look something like this. It's going to have something like this, and then it's going to start twisting. Okay, alpha helix, and then it's going to continue like this, and then it's going to be something like this. Okay, and then something like this, then maybe another beta, or whatever. The point of the fact is the polypeptide, the same polypeptide chain here, okay? This is the same, okay? This is the same one as, this was the same one as this one over here. It's just because now is doing hydrogen bonds with the between the polypeptide backbone. It's kind of folding in a different way now. Okay, so now we have a couple of alpha helixes. Then you're gonna have some beta pleated sheets. My apologies. And then we keep looking at the rest of the polypeptide. You're gonna see more alpha helices, more beta sheets throughout the whole polypeptide chain. So far, so good. Now, it's not just gonna stay like that. Now we're gonna have a tertiary structure. And the tertiary structure is also very similar, but this tertiary structure is gonna give you the final 3D shape of the protein. Okay, the final 3D shape of a polypeptide chain. Now, let me give you a call just so you understand the verbiage here. Many people say polypeptides, and proteins. So what's the difference between a polypeptide and a protein? Usually, different books have like polypeptide chain is just a chain, right? But when the polypeptide chain is ready, it's already folded, it's gonna be able to do its work. It's a functional protein. Then we call it a protein. Okay, but we can use them for our purposes. We can exchange this. We can use the same word. Okay, but just know if you go to another class, if you start reading other books, most of the times the polypeptide, when they say polypeptide, they just referring about the chain, the primary structure, maybe the way it folds. But a functional polypeptide chain is going to be mostly referred as a protein. Okay. Okay. So the tertiary structure is going to give you the final 3D shape of the protein. It's about all those twists and those folds that are the result, okay, of polarity differences between or different in different regions of the protein. So if you look at this picture over here, okay, this is gonna be like the interactions that are gonna lead to the polypeptide to the tertiary structure. Okay, and it's really determined by the variety of the interactions among the R groups. So if the secondary structure has to do with interactions between the backbone, the tertiary structure has to do with interaction of the R groups. Okay, so if you see this small little example here, I like it because it's showing you the different types of interactions that the R groups can have. For example, it could form ionic bonds, right? It can do it between an amino acid. This is one amino acid that I don't know if you can see it, but it has a positive charge and is being attracted to the other group, to this other R group of these other amino acid that has a negative charge. So do positive and negative attract each other? Yeah, they do. See, you see how the protein is interacting now, the polypeptide chain is interacting, but it has to do with the R groups. Okay, it has to do with the R group. Sometimes you're going to have, for example, another example here is you can have hydrogen bonding. Okay, but again, this hydrogen bonding is going to be between this amino acid, okay, and this other amino acid. So both amino acids, so these ones is probably going to be a polar amino acid with a polar R group, right? Polar R group. Um, what about over here? I want to do a different color. I want to do an orange color here. Well, here we have the interaction between this amino acid that is hydrophobic with another hydrophobic amino acid over here. Okay? And then remember I told you in the very beginning that in the introduction to proteins that sometimes you get to see sulfur. So sulfur, we do this in a different color. Sulfur is sometimes part of the R groups, and it can do something called a disulfide bridge. Okay, so this disulfide bridge can happen between two different amino acids, one amino acid having a sulfide group, another having another sulfide group, and they make something called a disulfide bridge. Okay, they bridge two things together. Okay, so you see how we made this tertiary structure, okay, with the interaction of the R groups with each other. Okay, now we have a functional property, a functional protein. Okay, that's all you need. Now, some proteins, not all of them, but probably most of them, but a lot of them are we're not done yet. Okay, some of them are made of one polypeptide chain, but then we have something called a quaternary structure. But this quaternary structure only applies, okay, when the protein is made of more than one polypeptide chain. And one of the main examples is hemoglobin. Hemoglobin is that protein that transports oxygen in your body, right? And this is hemoglobin. And you see that hemoglobin is going to have four different polypeptide chains. This is going to be number one, for example, number two, number three, and number four. So in order for hemoglobin to work, it is made of four polypeptide chains. Okay, four polypeptide chains. Each polypeptide chain is going to have its own interaction, its own alpha helices, its own beta plated sheets. It's going to have its own interactions between R groups. Okay, that's the way it folds that polypeptide chain. But sometimes a protein requires more than one polypeptide chain. Okay, forming a multi-unit protein. So I hope these structures made sense. One more thing about proteins, and it has to do with their function, right? It has to do with, I told you, in order for proteins to work, in order for proteins to work, let's say, correctly, they need to be folded the right way. That is the truth. But sometimes these proteins, if you put them in an environment that it just you put them in a different environment, you can unfold them. Okay, you can. It's called denature them. You can denature them. So it's a normal protein. What happens if you put it in an environment that is not the best environment for this protein? The protein can get denatured. It unfolds. And when it unfolds, is it going to have the right conformation to do what it's supposed to do? It's not. So a denatured protein technically does not work. Okay? It's no longer has the structure to meet its function. So protein unfolding and on the end denaturation. So a protein only works in its native or correct conformation. Changes due to the environment may cause this protein to unfold or denature. Some examples: increase the temperature so much. When you increase the temperature so much, you increase the motion that affects hydrogen bonding, okay, and the bonds between R groups. So remember we talk about water, you know how you have to put a lot of energy in the water to disturb those hydrogen bonds. So it's very similar, right? If you increase the temperature, the molecules start bouncing more, starts vibrating more, and that's going to affect the hydrogen bonding of the proteins. Therefore, they can denature. What about if you add them into a salty concentration? Well, because salt is an ionic compound, it could be ionic interactions between R groups. So it's going to disrupt these interactions between different R groups, and again, the protein is going to unfold, is going to denature. What if you lower the pH? Remember, if you lower the pH, okay, you're making the area more acidic. You're making the environment more acidic. And if that happens, if you make the environment more acidic, the concentration of hydrogen ions change, right? And this can affect how the protein interacts with itself or whether it interacts with the hydrogen ions from the solution or from the acid. So again, it affects hydrogen bonding. Okay, so for a protein to work best, it has to work at its optimal environment, the right temperature, the right cell concentration, the right pH, and then it's going to be able to work well. It's going to be a normal protein. What happens when it unfolds, when it denatures? The protein loses functionality. Okay, in some cases, it can, if you come back to the normal environment right away, proteins may refold, but many times if you don't, they stay unfolded, denatured, and you can no longer be able to use them for. It can no longer have its purpose or its function. One more slide. Sometimes proteins can be associated with other organic and inorganic molecules. And you will hear this when we start talking about a cell. Sometimes we have something called glycoproteins. So sometimes we can have proteins with sugars. Okay. A lot of the times these glycoproteins are going to be markers in the cell membrane of a cell. Sometimes you can mix proteins with lipids. Okay. And you make something called lipoproteins, proteins with lipids. And in the case, for example, of hemoglobin, this protein has to be able to pair up with a heme group. And the heme group is going to have iron, and iron really is going to be able to attach to the oxygen. Okay. And that, ladies and gentlemen, is what you need to know about proteins.